that people can have an organ donated right now? Like, what are the conditions in which that could happen? You guys know?
Go ahead and type in the chat windows, guys.
Okay, well, I mean, if you guys are going to say something, you can keep typing it, but I'll just move on and talk about it a little bit. So the ways that people can receive an organ right now is, first of all, if someone dies and donates their organs, and second of all, if usually someone who actually knows the person. In other cases, I think they've developed some systems so that people who don't know the person could maybe donate organs. But usually, if someone who knows this person, who's a family member, could donate an organ to them. But obviously, you can see if there's a problem with supply versus demand. There's a lot of people needing an organ transplant, and it's pretty hard for them to easily get access to organs. So there's thousands of people just waiting for organs. And sometimes, even if people have an organ that they might be able to use, it won't actually work for them, or it won't actually help them because of compatibility. So if someone else is receiving an organ from someone else's body, that organ has to have very specific properties which are similar to this person's body. Otherwise, your body might recognize it as a foreign object and might actually attack that organ and not let it function. So that becomes an issue. But one of the goals behind tissue engineering, one of the ultimate goals, even though there's a lot of other advantages of it, is to actually be able to generate a person's own tissues from their cells to sort of regenerate a person's tissues from their cells. And that would kind of eliminate the need for these people to be waiting for someone to die so that their organs can be donated.
So as we said, we already introduced ourselves, but I'm Jacob Quint, and my background's in mechanical engineering, and I screwed up Abby's background. So today we're going to focus on the cells and the methods used to generate and grow some of these organs and where we're at today in tissue engineering. So we're going to cover the cells that there are, that tissue engineers use to create organs, the environment that these cells have to live in, and how we are able to multiply them to kind of create mass-producible organs, and then kind of the scaffolding or three-dimensional structure that we grow these cells on. Then obviously the team of engineers that come together to work on producing these organs. So first we're going to talk about those cells that tissue engineers use. So our bodies are made up of several systems, as you guys may have studied in some of your biology or other classes growing up. And so as an organism, these organ systems vary. So we have all of these organ systems perform different functions throughout our bodies, such as our cardiovascular system pumping blood, skeletal system giving us support, skeletal muscle system allowing us to move, and digestive system allowing us to break down foods. And these systems are broken down of individual organs, which are comprised of several tissues that are organized in certain ways to give us those functions. And these tissues are made up of individual cells, and we're going to count those as kind of the fundamental building block. Now, as tissue engineers, we take these fundamental building blocks and actually work our way up through this hierarchy and allow us to recreate these organs. And there's several challenges on the way, but that's the ultimate goal. And so these tiny cells that we're using, we are able to manipulate them, but they're small little factories that dictate the growth and function of our bodies. And so they have different organs within themselves, like we have organs. And so they have a brain. It's almost like the information center, which is the DNA. It has all the information that's going to dictate how the cell behaves. It's going to be sending signals in and out of there, just dictating all the processes of the cell. And then you also have the mitochondria, which is like the power source. It's what gives the entire cell power. And you also have the ribosomes, which are tiny little organs that build little proteins. And proteins are actually the building blocks for our body. And it allows these cells to change shape and form structures outside of them. And then these cells, as I said, are always sending signals. They're sending signals in and out, almost like we constantly receive information on our cell phones. And they send signals to other cells and to their surrounding environment. And these cells really dictate how these cells behave. And so as you may have studied in your biology class, is that we all start from two cells. And so when these two cells fuse, we start to form an embryo. But from this embryo forms all the tissues throughout our body, from our bones, as I said, which give us support, to our muscles, which allow us to move our heart beating. And so from these few cells, all of these cells start to specialize into thousands of different types of cells. There's actually over 30 trillion cells within your body, and a lot of them perform several different functions. And so how did we come from just these two cells? Well, those two cells were actually called, once they fuse, they form stem cells. And stem cells are actually, we still have several in our body that we're going to talk about, but stem cells are any cells that can start to become any of these specialized tissues. Now, there's a whole bunch of different types of stem cells, and some can become certain types of tissues and not others, but generally a stem cell can become several other types of cells. And the benefit of having a stem cell as a tissue engineer is it allows us to form whatever tissue type we want, and we also allow them to be multiplied multiple times. If you just take an adult specialized cell in the body, it can only be multiplied several times. But these stem cells can keep being reproduced, so we can keep multiplying them to make actually economical amounts of cells, so we can kind of make tissue engineering a reality. And so stem cells, as I said, we know what they are, but where do we find them? How do we get them to be used for medicines? And so there's several types of tissues that we're finding these stem cells in, and scientists are finding them in new tissues all the time. And so they found them in muscles and in brains and within your skin. And so, as I said, you only have multiple times to reproduce these cells. So, for example, when you're working out, you need to regrow muscles after you've damaged them, or as you're growing, you need stem cells to help your brain continue to grow, or your skin, if you get injured, you need to be able to keep healing these tissues and regenerate them as you get older. And that's what the stem cells are doing throughout your tissues all the time. But the stem cells in these tissues are relatively hard to get to. And so engineers work together as a whole, and we've found some sources that are easier and more accessible through surgeries, and those are in your fat. So if someone's undergoing liposuction, they can filter out your stem cells, or they can go into your bone marrow. And within your bone marrow, and kind of this spongy material here, that's actually where a lot of stem cells reside, and they can put a needle in there and draw out some of those cells, and then those can be mass produced and used for several different tissue applications or even medical applications that we're using today. And then?
All right, now I'm going to talk about sort of the environment that cells need to be able to grow, because cells do require, much like humans, a very specific environment, and we have specific needs in order to actually grow and thrive to the scale that we would need them to in order to develop tissues. So cells need very similar things to survive that we do. One of them being nutrition. They need different nutrients from some kind of food, essentially. Cells also need oxygen. Cells also have to be kept at a specific pH level. If cells are at a really high or a really low pH, they will die. Their proteins will denature, change their shape, and lose their function. So cells need to be around a pH of 7, actually about 7.4 for human cells. Growth factors are signals. So different signals tell a cell what to do. They enable its behavior. They tell the cell, the stem cells, what are they going to turn into and whatnot. And cells also need to be kept at a warm temperature. And finally, waste actually needs to be removed from the cells. Much like at a large scale, organisms have waste that need to be removed. On the small scale, cells also have waste that needs to be removed. So engineers will usually grow cells in a flask, which is the plastic container that you see here. And this flask contains the cells.
Lost Abby for a second. In this meantime, I will fill in. I forgot one thing about stem cells too. There's actually a recent technology where they're able to take any type of adult cell, so any one of those specialized cells in your body. Oh, you cut out for a second, Abby.
Oh, I'm sorry. Am I back now?
Yeah, do you just want to start with this slide again?
Oh, I'm so sorry. Okay. So as engineers, we want to recreate the environment that the cells need to grow. So usually we grow them in a flask, which is this plastic container that you see. And this flask contains the cells, and it also contains what we call media. So media is this liquid that the cells live in, and it has medicine, it has the cell signals that we were talking about before, growth signals, other signals that the cells need to carry out their functions, and food for the cells to use to grow. Its pink color comes from the pH responsive dye, And this will alert scientists if the dye changes its color, which it will at a different pH, that the cells are becoming unhealthy. And then the scientists can try to respond to that and help the cells. The cap of the flask has holes in it so that the cells can remove carbon dioxide and take in oxygen. And every so often, the cells need to actually be washed, so their waste is removed and then new media is put in. And they're also usually kept in an incubator, which keeps them warm, and it also keeps the concentration of gases that they need surrounding them at the right levels.
So next, we're going to talk about how do we take these multiplied cells, as Abby just covered, the environment they need to grow in. So they're growing in a two-dimensional environment in that case. How do we kind of put them on a larger scaffold and form them into three-dimension? So that's what we're going to talk about now. So in order to form three-dimensional tissues, we need to create some kind of supportive structure, and we call those scaffolds for the cells to grow on. So once again, the overarching idea of what organ transplant and organ donors are going to look like in the future is that we're actually going to be able to take human bodies, who either are healthy or even diseased. We can take those cells and turn their cells into stem cells, and then we can grow them and multiply them into a dish. And then we can provide them that three-dimensional supportive structure that engineers are working on building now. And then we can combine them with those cells, and then we can grow them in those artificial environments and then implant them back into that same patient or somebody else with an identical immune response system. So scaffolding is a very important concept in tissue engineering, and you may have heard of scaffolding before. And this is a building on the right here that actually has scaffolding on the outside. And so scaffolding is used when building buildings to give them some kind of supportive environments for workers to rebuild or even construct buildings from scratch. And so these are temporary structures generally just during the building of the system. And that's kind of what the tissue engineering scaffolds are trying to do too. And you see here, these are actually scaffolds for ears. And so what the scaffolds do is they provide a really nice supportive structure for our tissue engineering constructs, and they actually allow it for form. So if we didn't have this structure underlying it, the cells would just form this really blobless mass that would not really have any real use. And so one of the key components of the scaffolds is it allows the structure to organize these cells in a really unique way. So if we're using multiple cell types, we know how to constrain all of the cells within these scaffolds. Another thing we need when we're building a scaffold, besides just the structure that's there and how we organize the cells, is we need it to be porous or open air. And you can see some porosity here, but we'll show you another picture here where you can see a scaffold with a lot of porosity throughout it. And this porosity is very key because it allows those cells to grow into the scaffold, but also to have their waste and nutrition flow in and out of the scaffold. And that's one of the challenges we currently have, is making really thick and really big full tissues. You actually need blood vessels which do that for you. And we're kind of limited on that, and those are very detailed. So we need porous structures, but we also need them to continue to be temporary. We don't want these scaffolds to be permanent, because we want these scaffolds to actually be degraded by your body so that the new tissue can form around it and form in a natural way. And as these scaffolds start to degrade, we can actually use them not only just for the form and structure, and we need them to degrade, but we can actually embed them with chemical signals to tell these cells what to do, as Abby was saying. So you can take these stem cells and tell them what kind of cells to be into. And even beyond chemical signals, based on how hard and the mechanical properties of these scaffolds, if you have a hard scaffold and put a stem cell on it, stem cells are more likely to turn into certain types of cells, such as bone cells. Bone cells like really hard and stiff scaffolds. So how do we make these scaffolds as engineers? Well, some of the oldest methods they've been using is actually taking organ donor tissue that couldn't be used in a patient because of immune response. They're actually able to strip the cells off using a special washing method, and they're actually able to decellularize it. And so this is actually a rat lung that they're able to decellularize. So they take out all that genetic information and everything that makes that rat lung unique to the original rat, and then they're able to actually infuse it with new cells and then implant it into a new organism who needs that tissue. But the problem is that we already have the limitation of organ donor tissue, as Abby said. And so how do we make these from scratch in a very detailed manner? We don't carve them or mold them. What we actually are currently doing is using 3D printing to make unique structures. So this is a little time-lapse video. And so here they're actually laying down a biomaterial in a unique porous scaffold material. And this is just going to be acellular, which means there's no cells in it. But currently they're working on optimizing this process by including cells in it too, and several cell types or several material types, so you can create very detailed and organized tissues that have special roles. And so the finished products of these tissue engineered constructs are really cool. And a few of these are even being used in people right now. And so this is an ear, as I've showed earlier, where they're able to seed the cells and then grow it in that environment and then plant it into somebody. So if you have a deep birth defect where you weren't born with an ear, or if you get into an accident, they can replace it with one of these constructs. A really big one that's being used right now is skin tissue. And so they've actually been able to engineer skin for people who suffer from burn injuries or other accidents. And these two are really being used along with bladders. Those are some of the very few tissues that right now are good enough to actually be used in patients. But tissue engineers are becoming more and more aggressive every day and trying to create the coolest and next technology to help medicine. And they've even created models and little artificial hearts. And so these actually have cardiac cells in them. And this is a little heart that's been printed. But they're also trying to give them function. This one, the cardiac cells on their own will beat within this little heart, but there isn't the contraction you see in a whole scale heart. But what they're trying to do is give things function. And this is actually a little lung here that actually is able to perfuse oxygen and pump it. And so it models the lung. And this is actually a really short snippet of what these little lungs do. There shouldn't be any sound, so don't worry if you don't hear anything. But it's actually able to beat. And it's actually able to pump blood, which is pretty cool. Ooh. Oh, now it's just flipping through. Okay.
All right, so finally I'm going to talk a little bit about the role that engineers play in making all of this happen. Tissue engineering is really difficult. Obviously, we're kind of in the early stages of it, and we need a lot of different people with a lot of different really specific backgrounds and expertise to work together in order to make tissue engineering happen. So first we can talk about mechanical and electrical engineers. Remember that we talked about the 3D printing part of the process, and we talked about the incubators that the cells will live in. So mechanical engineers sort of work with the mechanics of all that and how they work, and electrical engineers would be responsible for how those are powered and things like that. Chemical engineers are going to know things like what material can we make the scaffold out of so that it will metabolize and so that it will decay as the tissue grows so that it won't be stuck in there. And they would also know, for instance, what signals and what molecules should be sent to the cells to tell them to differentiate into the cells that we want them to. Biological systems engineers would probably be intersecting a little bit with all those different other areas, but they mainly would actually know the most about how the cells really work, and so they would be able to inform all the other engineers about how they should create their machines and their structures in order to best serve the needs of the cells. So maybe the others would know how to do something, and the biological systems engineers would be more informed about what actually needs to be done, if that sort of makes sense. So all of these different engineers have to work together in different ways, which I kind of just scratched the surface of in order to make the tissue engineering actually function. Anyone have any questions?
You can ask them by unmuting, or you can type them into the chat window if you have questions. We can probably move on to the activity.
That's good. So I've been informed that you should know this engineering design process by now. So we're going to make it more of a little bit more of a review. And so maybe unmuting would be the quickest if you have a response to this. But does anybody know what the ask portion of engineering design process is, or maybe even the next thing? No responses. That's all right. We can fly through it. So with the engineering design process, you first want to try to isolate the goal that you're trying to do. And so you should ask yourself, what is my goal? What are the questions and kind of the assumptions that I need to know in order to solve this problem? So today we're going to be covering how to create a heart valve, and we'll talk about how that relates to tissue engineering in a bit. But you have to ask, okay, perhaps in this situation you need to be like, what are my limitations? What are my constraints? What kind of materials can I use? As Abby said, you can't have any materials that are adverse to the body, and so we're limited there on what materials we can use, but not in this case because you're using a cardboard box, so there's no bodies involved. But then you have to start to imagine what your solutions would be. Based on the constraints and the information you found out in your asking phase, you then will be able to imagine the solutions in your particular heart valve. Next, you're going to start planning. Now, usually as engineers, we spend months in fine-tuning our plans and writing down ideas and sketching them out and constantly improving them as we'll get to, but you have to plan thoroughly to ensure that your plan to achieve your goal is actually going to happen. Then you have the fun part. That's where you actually get to create your solution and try to build your actual idea. And then you have to ask yourself again and improve your solution. You have to ask, how can this be improved? How can we continue to improve our project? And you're going to have to do this multiple times. We call it iteration in engineering. And especially today with this somewhat challenging activity, you have to continuously improve your design, even if it's really quick. You're going to always be improving and then working your way back through this design process. So today we have a heart valve activity. So hopefully you're going to be saving a life with marbles. And so the heart is a really important muscle in our body, and it actually has different valves that control the flow of blood. And so we actually pump our blood from the unoxygenated portions of our body through the lungs, oxygenate our blood, and send oxygen and nutrition, excuse me, to the rest of our body. One second. Ooh, sorry about that. But we also have these valves that allow for the control of blood flow. And so there's actually several different diseases that don't allow for the proper functioning of these valves. And so some children are born without these valves, as I talked about with my undergraduate research. And so then they actually have to have them surgically replaced, or if you're older and have a heart attack, or if you have some other malformation of your heart, you have to get these valves replaced. And so they can use mechanical valves. You can use animal tissue valves, but both of those have limitations and there's complications with them. And more recently, we've really been investigating tissue-engineered heart valves that actually have the patient's live cells in them. And those actually combat a lot of the challenges with both of these. And so this, even though you may not see a heart valve as tissue engineering, that is the future of tissue engineering, is creating things like this. So today you're going to use this design process and be biomedical engineers. And you're going to go through this design process and how you're going to replace a mitral valve for a heart. And so you're going to work by yourselves because of coronavirus. And you're going to have a shoebox that's going to represent the two chambers of the heart. And I'll go over that in the next slide. And you're going to design a valve to go between those two separate chambers of the heart, much like it does in the heart. And you're only going to allow your marbles to flow from one of those chambers to the other, but not backwards, because that's actually called backflow in our heart valve. And we actually don't like any bleeding. You don't want any of your blood to flow backwards in your heart, because then it's not getting pumped to the rest of your body. And so you want it to be a one-way valve. You want the marbles to go through but not come back. And it must be freestanding. You can't hold it. Sally, are you going to let them tape it to the box because it's their own box? Or
you can tape it to your box, but if you want to make it a bigger challenge, then try not to attach your valve to your box. So we don't let students attach them to our box because we reuse our box. So it depends on how much you want to challenge yourself.
And so before, you all may have some marbles, but you're supposed to, once again, only be able to test with one marble before we run our final test with a whole bunch of marbles. Your box will behave a little bit differently with all the marbles in there versus one. Think of them running into each other. But you should have one marble you can continuously test with and keep improving your design. And so you're going to create a model heart out of a cardboard box. And so you're actually just going to have the before picture of your cardboard box. And then you don't have to spray paint it red as is shown here, but you need two little leaflets that kind of allow to separate one side of your chamber of your heart to the other. And then you put your valve right here. And then, so the materials you're going to need, I think Sally's kind of told you what you need. You're going to need at least a box and then some of these materials to be able to build your valves. And then, yeah, if you don't want that challenge, you can build the box. Is there a video you want to show or anything, Sally?
No.
Okay, sounds good.
The video has some solutions, so I don't want them to see the solutions. So we're going to cut out pieces of cardboard. We're gonna prepare our boxes. Get my marbles out.